going. All right, there we go. Waiting on it to start up, make sure it works. Hello, biology students. Welcome to your student notes. This is our third part of learning about DNA. Yesterday we should have wrapped up replication, where DNA makes a copy, an identical copy of itself, and that's used for when cells divide later on. So they'll have the exact same copies of DNA. Today we're getting into something really cool and really unique. One of the first things you learned about DNA was DNA uses RNA to make proteins. Okay? DNA uses RNA to make proteins, and proteins help carry out all functions of life. As humans, it helps us make us who we are. It also helps all of our organs work correctly. Little proteins come together and they cause everything to function correctly. DNA, of course, being the blueprint of life, it needs to get the message out what proteins need to be made, how many, where are they going to go, what are they going to look like. DNA is going to use RNA to make these proteins. All right, before we dive into this process of transcription, I wanted to give you some terms to learn here. Francis Crick of Watson and Crick fame defined what we call the central dogma of molecular biology. You can think of it as the big rules, the central rules of molecular biology. At one is replication copies DNA. That's the process we saw yesterday, where DNA splits apart. We got our DNA molecule, and it's going to rip completely in half, and then nucleotides are going to fill in and cause it to have two identical, imagine if I had one here, identical copies of DNA. That's replication. Transcription, what we're going to cover today, is how DNA makes a message into an intermediate molecule, or a molecule that's going to carry the message, called RNA. All right? And then into next week, we'll learn about a process called translation that reads the RNA message and causes the message to be kind of understood as these amino acids which come together to make proteins. Think about if you and a best friend or something had a little secret language, right? If you kind of had some special words that meant special things. You were writing a note to somebody and you didn't want your parents or your teacher or whoever to see the note. You would transcribe or write out this message using only the language that you and your friend knew, right? So you would write it on this note, and then you would hand the note to your friend or whatever, and if somebody intercepted it, they may have some trouble reading it, but if your friend got it, they in turn would look at this message with this code on it and translate it and be able to understand it. So transcription, think about it this way, is how DNA writes its code. Okay, it's writing its code to hopefully, in the end, make proteins. All right. In prokaryotic cells, remember those are very simple organisms that do not have nuclei. This takes place in the cytoplasm. We're really worried about eukaryotic cells, like what's in us and what's in advanced organisms. Replication, what we learned about yesterday, and also transcription, it takes place in the nucleus, where the DNA is. RNA, as we're going to see, is able to leave out of the nucleus into the cell itself, into the cytoplasm, and that's where translation is going to occur with those special structures called ribosomes. We'll remember all this as we go, hopefully. All right, so RNA, this is the new thing we're learning about today. It acts as a messenger between the nucleus, or the DNA that's in the nucleus, and protein synthesis, or the making of proteins that's going to happen in the cytoplasm. RNA differs in three main ways from DNA. Okay, so DNA, we kind of understand it, a little bit about it. Now, how is it different? RNA different from it? Well, one, the sugar is different. Remember, deoxyribose is the sugar in DNA. It's how it gets the name DNA, deoxyribonucleic acid. RNA stands for ribonucleic acid because its sugar is ribose. So ribose, ribonucleic acid. All right? In RNA, there's a slight difference with the nucleotides as well. It has one of these bases, or a nucleotide, called uracil instead of thymine. We've learned all this about apple in the tree, car in the garage. Whenever we have DNA, A and T are always going to pair up according to Chargaff's rules, and C and G are always going to pair up, right? Well, here's the thing. With RNA, there is no thymine, all right? So this is going that kink into it. If we have RNA, it'll instead have this uracil. So we'd have A pairing up with U, and C and G still pairing up together. So car in the garage is still good. But A pairs up with U in RNA. That's the only little difference about those. So instead of apple in the tree, it's apple whatever you want to come up with, okay? So adenine is always going to pair up with uracil. Another huge way that they're different is RNA is a single strand, not a double strand. Double helix, it's got these two strands, 1, 2. It instead is just a single strand, as we're about to see. Okay, so RNA, different kind of sugar. It has uracil instead of thymine, and RNA is a single strand, not a double strand. All right, that's the basics of what you need to know about the differences in RNA and DNA. Transcription, or this writing of messages, is actually how we get 3 different types of RNA. Transcription, as we're going to see, copies the sequence of DNA, or one little gene, or one portion, and it makes RNA polymerases, as we call it. This is an enzyme that helps make RNA. All right, so what happens in the whole process of transcription is DNA is going to begin to get unwound at a certain place, okay, where a gene begins. So just like we saw in replication, the DNA strand is going to break apart at those hydrogen bonds in the middle. Instead, this time they don't break all the way apart. They're just going to temporarily separate, okay? Temporarily separate. Nom, nom, nom. Then what's going to happen is, using one strand of DNA, so I'm just going to lay this back one over here, okay? It's still attached, but we're just worried about these 3, sequences here. Complementary strands of RNA are going to be produced. And the way they do that is this polymerase, this enzyme is going to come through it. Check out this animation. It comes through, it unzips the DNA molecule just like I've done on mine. And what it's going to do is, you can see, well, I'm pointing at it, but as you can see, go back one. All right, it unzips it temporarily, and then you can see these free-floating nucleotides that are found inside the nucleus. They're just kind of hanging out. They're not attached to anything. They're going to come in here and start filling in some gaps along that red line. Okay, they fill in these gaps. And as the polymerase goes through the DNA, unwinding it, it's going to leave this long chain of these base pairs that match up. Okay, G is made because it connected with a C, whatever that may be. The U is there, uracil, because it had matched up with an adenine, an A and U, remember? C, of course, it matched up with a G. And what we have is this long coded message. It takes this gene that we find in the DNA molecule and makes a message out of it. Now, it's a message that basically contains that DNA message, right? Because it's just writing the opposite side of the DNA, except we have uracil in this case. Okay, so it detaches, and then the polymerase goes away, and we're left with this nice little strand of mRNA, as it's called, or this strand of RNA that's a messenger. All right, so the DNA code, this trillions of base pairs long, what it really is this message to make RNAs, because we're going to see later that these RNAs are what's going to be used to make proteins, which enable life. So from this process of transcription, let's just look at it here before we jump on this slide. Remember, you can pause this video as much as you need to or go back. It is, there for your benefit. So we've unzipped it, right? Okay, I know it's a little hard to see, but we've unzipped, and we've got these 3 here that are ready to go. In the nucleus, there's these free-floating nucleotides. La, la, la. The polymerase has come through, unzipped it. What's going to happen naturally is these nucleotides are going to come in and pair up. So if this was cytosine or C, this would have been a guanine or a G, okay? So if this next one, would have been an adenine, let's say, then you guessed it, a uracil, okay? Because that's RNA. We're making an RNA strand. That uracil would have came in and connected here. All right, next, if we would have had, I think this was another cytosine, we would have come in and made another guanine. Now, if this was the end of the gene, if this was all that the message needed to be, what's going to happen from that point, is now this RNA that's made these covalent bonds here, right? And these are strong bonds. These are hydrogen bonds in the middle. Those are weak. These covalent bonds, they're going to hold together. And now we have a message. We have RNA ready to go. So, it's going to go out into the cell to make a protein. Meanwhile, that polymerase, this is the other side of the DNA, is going to attach it back together. Okay, and what are we left with? Our original strand of DNA. Hello. Original strand of DNA. ready to make more messages as it goes. Now, this whole process of transcription not only makes messages, messenger RNA, it makes 3 types of RNA. Now, the main one we're really focusing on right now is messenger RNA. That is what we have here. It carries the message of DNA for what proteins need to be made. So mRNA, the little m stands for messenger. This is the code, wink, wink, code. Your quiz probably says code in a little bit. Messenger RNA. It also makes ribosomal RNA, and this makes little ribosomes later on. Okay, these are made in the DNA. It goes out, it makes some little proteins that come together to make ribosomes. It also makes something called tRNA, or transfer RNA. And we'll see how this is huge in translation, that third process of Crick's central dogma, remember, in that first slide for today. This transfer RNA, as we're going to see, it helps bring amino acids together and makes these proteins, okay? So we're really worried about transcription, though, makes the message. So let's think about it. How is it similar to replication? Both of them occur in the nucleus. That's where it's occurring, in the middle of the cell. Both of them involve that DNA coming apart, okay, those hydrogen bonds being broken, and both of them involve a complementary base pairing. Whereas replication is coming in and it causes 2 identical strands of DNA, and this one is coming in and complementing it with this this RNA strand, which then will leave the DNA molecule and go out into the cell. Okay? So let's just check here for understanding. Hopefully you're filling in your student notes and it makes a little sense. Of course, I got some videos to help make a little more sense of it today and next week. What's the central dogma for molecular biology? 3 tenets of it, 3 points of it. 1, replication makes identical copies of DNA. Transcription helps make RNA, okay? It makes the messages. Translation, as we're going to see next week, reads those messages and makes proteins. DNA uses RNA to make proteins. Okay? Where do these following processes take place? Replication and transcription, both of them in the nucleus. Translation, as we'll see, takes place out inside the cell called cytoplasm. 3 main differences in RNA: the sugars, deoxyribose versus ribose, the single strand instead of a double strand like we find in DNA, and the RNA uses the base uracil just in place of what we've learned to be thymine. Okay, so adenine, A, always pairs up with uracil, U. Okay, 3 types of RNA: mRNA, which stands for messenger RNA; rRNA, sounds like I had a little stutter there. rRNA has ribosomal RNA, makes up ribosomes, and tRNA, transfer RNA, which we'll see is a huge role in translation. Okay, and then number 5, how are they different? Hopefully you understand replication is making identical copies of DNA. We wind up with 2 identical strands of DNA. Transcription, of course, is writing, if you want to think of it that way, the RNA. Okay, it's going to carry this message out into the cell to make proteins. DNA uses RNA to make proteins. All right, feel free to watch this as many times as you need to until you got a grasp of it. Feel free to use your student notes as well as this video, taking a little quiz today, and then I think you have another little video to do today as well. Sitting here.